Welcome to class, my friend. I am here with my brand new microphone. So, here's hoping that it sounds litty as hell. Oh my god. It just wouldn't be it wouldn't be lecture day if I didn't have a host of technical difficulties plaguing me. Sound good? Thank you. Thank you. I'm glad. This is the first time I've had to use like two entire different devices to use a mic. I have a preamp and I can't lift it because I'm afraid that my house of cards is gonna fall, but I have a fucking it's called a lifter because the microphone is too soft. They just make things up for us to buy. But in any case, I'm here and it seems to be working. But hell yes, I was hoping you would be. Uh Gretchen specifically told me that she was going to the dispensary today in order to prepare for this journey that we're about to take. um I feel like probably everyone here uh is pretty well versed in weed and THC at least, and maybe even a little bit of CBD. I think if you went to the college that Kurt and I did, then you certainly you have a passive education about all things cannabis. Oh, am I a little quieter? Okay, that was a little test. Um, oh, I mean I know you don't weed, but if you anyone who goes to our school is inevitably bombarded with all things cannabis and THC, and a bunch of people being like, "Oh my God, you have anxiety? Just smoke weed. It literally solves everything." Um, it's like, nope, weed school exactly. And you would think that the students of weed school would know, hey, weed doesn't really cure everyone's anxiety. I actually, when I worked at the dispensary, I used my biology degree more than I did when I was working at like labs and institutions. Like I was more inundated with like really cool questions about how it worked, and we would have vendors come and sort of explain to us the mechanisms, and for the most part they were really kind of regurgitated bullshit, but you would find some very interesting things in there. So I'm hoping that despite how flooded we are with talk about cannabis and THC and CBD, I hope that I teach you something today. Yes, I remember, and I remember there were pictures circulating of the only people to get arrested at 420 in Santa Cruz were, I think, these two guys who made a pound joint, a pound into one joint. And the cops were like, "You know what? That's a bit much for us. That's we're going to put a stop to it right there." Yeah, yeah. And yeah, it was so nuts. People would, oh my god, flying in, which means you have to fly into San Jose and then take Highway 17 to get to us. It's like you could go to San Francisco. There's like an airport just south of there, but no, people wanted to go to fucking Santa Cruz and go to the meadows on a college campus to celebrate. Oh man. I uh I never I didn't try until after I graduated. Uh yeah, like literally like the summer after, because I took an extra uh quarter, but man, there were some people even in my major who would like get stoned all the time, and I'm just like, how do you do that? There's no way. There's no way that I can do this and then study. But they a 2-pound joint. Oh my god. That is like, I mean, that's a waste if anything. I mean, wow, that's fucking crazy. Let me look at that. Oh my god, please say they have the picture. Yes, yes. Oh my god. That fucking cop. Oh god, it's so hilarious. It does. It looks like a fucking boa constrictor. It looks like it's gonna choke me. um, well, as we're about to find out, I am mostly going to be focusing on the THC that lies within us, our endocannabinoids. Endo, of course, meaning inside, and cannabinoids meaning uh, coming from the cannabis plant. Um, so, what's up, Saga? THC is it's that stuff that gets you high when you smoke weed. Uh, it's I actually don't know if I can pronounce it after all this time, but and I don't know what it is in Spanish, but in English it's tetrahydrocannabinol. It's ooh, heck yeah, I can't wait for your friend to get here and be so disappointed. I'm just kidding. I'm going to blow their minds. But yeah, for anyone who doesn't know, I know I just talk shit and I talked about how everyone knows THC, but if you don't, it's the psychoactive component inside of cannabis. There are a lot of other ones, but that's the one that we know the most because it makes us high. Um, and we're gonna look at the functions of the cannabinoids that our body makes itself and how they comprise what's called the endocannabinoid system. So we're gonna fucking get into it. Interesting. Okay, so the endocannabinoid system is a group of it comprises different parts of the brain and other areas of the body, usually areas that secrete hormones like different glands, as well as the GI tract. It's technically everywhere in your body. All right, I have notes. Cock and cock and ball torture. I'm I'm so upset that my brain didn't see that before, especially since CBT is cognitive behavioral torture, um, or cock and ball therapy. But now I won't be able to unsee that. Jesus Christ. Okay. So we already have the system in our body that's composed of these cannabinoids that we use to get high. And what endocannabinoids are just fatty acid derivatives that act as retrograde neurotransmitters. So neurotransmitters, they are in our nervous system. They are released between neurons in order to facilitate communication. And retrograde literally means that they act in reverse compared to other normal neurotransmitters. Um, and broadly, what the system does, I'll explain retrograde neurotransmission in a little bit, but broadly, what the system does is it regulates anxiety, the immune system, memory, metabolism, pain, sleep, stress, and I forgot to put this in the presentation, so I'm just going to remember it later and explain it. Embryo implantation. So weird. So all of these are obviously very important, and it's quite an eclectic group of functions that the endocannabinoid system has, um, which demonstrates just how vital it is to our existence. All right, so what makes endocannabinoids different from pretty much every other neurotransmitter is that they're made on demand. Pretty much all other neurotransmitters are made in advance and then they're stored in vesicles until it's time for them to get released. So the fact that they're made on demand means that they can mediate pretty immediate effects, especially in the brain. Um, and broadly, we're kind of looking at this here. When a neurotransmitter gets released, it gets released by a presynaptic neuron before the synapse. The neurotransmitter then traverses the synapse, which is just a space between them, and then activates its receptor on the postsynaptic cell. It's pretty simple. That's pretty much basic neurotransmission. But how endocannabinoids are different are, instead of getting released by the presynaptic cell, they get released by the postsynaptic cell in response to some sort of stimulation, such as another neurotransmitter binding to it. And then they get released into the synapse and they travel backwards to the presynaptic neuron. And that's where they bind their receptor. And the end result is that they somehow affect neurotransmitter release. Usually it's inhibition. Usually, let's say like this neurotransmitter is dopamine. An endocannabinoid would bind on this presynaptic cell, and the result will be lower dopamine release. So yeah, this is retrograde neurotransmission, and this is how all endocannabinoids mediate their functions. And it is important to note that mostly what we're going to be dealing with, we're not going to be dealing all that much with dopamine, unfortunately, but we're going to be dealing with these two neurotransmitters, glutamate and GABA. And I believe that I have brought them up in every single lecture that I've done. They're vital to our brain's existence and to our ability to have these connections within our brains that keep us alive. So I'm gonna say glutamate and GABA a lot, but it's just important to remember that glutamate is the excitatory neurotransmitter. If there is glutamatergic neurotransmission going on in a brain area, then that means that that area of the brain is active. There are lots of electrical and chemical signals happening there. Neurotransmission that is mediated by GABA, and that is GABAergic, is the opposite. That is the inhibitory neurotransmitter. So it dampens any sort of communication, any electrical activity in specific brain areas, depending on if it's being released. So that's going to be a big thing to remember throughout the rest of this, is the difference between those two guys. Yeah, this is fine. That being said, you know that endocannabinoids act on presynaptic membranes to lower neurotransmitter release. And now you know that there's an excitatory, a dedicated excitatory and a dedicated inhibitory neurotransmitter. Obviously, things are going to look a little different depending on where the endocannabinoids are acting. For example, on the left, we have a glutamatergic neuron. It is excitatory. Usually, it facilitates communication in the brain. If endocannabinoids bind on a glutamatergic terminal, they're going to lower the amount of glutamate being released, and the net result is going to be inhibition. It's going to be a lowering of that communication. On GABAergic terminals, it's going to be the opposite. We're going to see something called disinhibition. So endocannabinoids bind on the terminal, they lower the amount of GABA, the inhibitory neurotransmitter, and the end result is going to be excitation, because you have removed whatever inhibition is being placed on any given postsynaptic neuron. So what type of effect happens as a result of endocannabinoid binding is entirely going to depend on what type of neuron it's binding to. And as we can see on the right, cannabinoid receptors, I believe this is CB1, they are mostly going to be expressed on inhibitory neurons, meaning that much of the time, if something is activating a cannabinoid receptor, it's probably disinhibiting something. It is still expressed at a great deal on excitatory neurons, but there is sort of a discrepancy between the neuronal types that the cannabinoid receptors are expressed. And speaking of the receptors, I have there are a couple that I might like passively mention, but there's two main cannabinoid receptors, and that's going to be CB1 and CB2. Now, CB1 is pretty widely expressed. We see a lot of it in the central nervous system. We see a lot in the placenta because of its function in embryo implantation, which I will totally remember to talk about. But it is mainly a receptor that is expressed in the CNS. And I'll tell you right now that this is the receptor that THC likes the most. Oh, and fat tissue. That's important too. It's in fat tissue. I'm going to go in depth into cannabinoids when it comes to like feeding behavior and metabolism, but I did want to note that for whatever reason, infertile males possess less endocannabinoids in their sperm. So we could see here AEA is an endocannabinoid, as is 2AG. Fertile sperm, especially in the semen, fertile sperm have a lot more endocannabinoids in those areas, sort of denoting that endocannabinoids in some way mediate specifically male reproduction. Just a little fun fact. And then here, these are so FAAH is an enzyme we'll see that breaks down endocannabinoids. So there are all of these are enzymes that are in the biosynthetic pathway for endocannabinoids. So it's a little somewhat disordered in fertile sperm, it seems. And then if we look especially at the receptors, there's significantly less receptor expression in the sperm of infertile individuals versus fertile individuals. I don't know the mechanism behind this. I just thought it was, I just came across it and I thought it was interesting and it had the numbers. So I learned something about endocannabinoids in sperm. This is the main stage here, though, is the brain and the central nervous system. We have a lot of cannabinoid 1 receptors here, especially in the well, the cortex doesn't count because that's the big part of the brain. So whatever. We care about the hypothalamus and the basal ganglia, as well as where's the amygdala? The amygdala and the hippocampus. Those are brain areas that are going to come up a bit when I talk about the more specific functions of endocannabinoids. But this is the pattern of expression. But yeah, especially the amygdala and the hypothalamus. I'm really going to zero in on those guys. Yeah, I had a really, it was just a fluke that I came across that stuff on male reproductions. Although what could have happened is I think I saw like, whoa, why is there so much expression in the testis? And I looked it up and yeah, it's interesting. Okay, so CB2, not super common in the central nervous system. But that's okay because, oh my god, it's pretty much everywhere else. Especially again, those endocrine tissues, endocrine glands, the gastrointestinal tract, various muscles, and a lot of immune system structures. As we'll see, CB2 is going to be important for mediating certain immune responses. Speaking of the immune system, the most common place in the body where we find CB2 receptors is B cells, which are part of the adaptive immune system. They make antibodies and they present antigens. And endocannabinoids, particularly 2-AG, are needed in order for them to have their correct responses. I usually type up my notes beforehand, but I didn't have time, so they're all handwritten, and I cannot read my handwriting, and I hate myself. But that's okay. These two receptors, like, there's technically more cannabinoid receptors, or receptors that will bind to cannabinoids, but these are the two proper, like, cannabinoid receptors proper. And despite the fact that they are very, very similar in structure, they obviously do very different things depending on what is binding to it and where it is located. So we're gonna do some hardcore molecular biology right now. So get ready, because both cannabinoid receptors are G protein coupled receptors. And that just in a literal sense, that's just what it means. It means that the receptor is coupled to G proteins, which are these 3 different subunits right here. We call them G proteins because they bind to GDP and GTP. In fact, this should be a GDP right here, but that's okay. Specifically, the G protein that they are bound to is GI/0, which is the inhibitory G protein receptor complex. I know I've talked about before the GS coupled receptors, and those guys end up increasing the level of cyclic AMP in cells, cyclic AMP being a second messenger that drives signal transduction. But in this case, the opposite is happening. We are seeing a lower amount of cyclic AMP being made as a result of an endocannabinoid binding to these receptors. And which makes sense with the more over the surface function that I described earlier of endocannabinoids kind of dampening signals in neurons. So this sort of underlies that function that it has. And so it is this decrease in cyclic AMP that leads to decreased neuronal excitability. Remember that okay, I can read. And it's especially important here because I put animations, which means there's a bunch of cues, which means I have to press the button at the right time. I'm just going to really quickly go through the mechanism here of what exactly happens when an endocannabinoid or cannabinoid binds to one of these receptors. So one of the ways that neurotransmitters directly decrease neuronal excitability is by increasing the activity of potassium channels. And when there's a protein called PKA bound to these channels, they are not active. So, right, that's what we have right here. I have a PKA, protein kinase A, is bound to the potassium channel, and it is preventing it from opening. So it is not functioning as a channel right now. Man, I'm just gonna wing it because I cannot read my handwriting. Okay, so our potassium channel is not doing jack shit right now. And we'll understand the importance in a bit. But normally we have something called adenylyl cyclase making all of our cyclic AMP for us, and keeping this second messenger neurotransmission open. And those cyclic AMPs actually bind to the regulatory subunits of PKA. What you see right here is an inactive PKA because these regulatory subunits are bound to it and preventing it from having any action. So when cyclic AMP binds to those things, they change shape, and this shape change releases the PKA in its active form, and then it goes and it binds things like potassium channels to inactivate them. And again, this is all because we have cyclic AMP hanging out that is making this happen. So obviously when an endocannabinoid binds the receptor, something a little different is going to happen. These shape changes are going to cause the subunits to go and have their own little functions. So the alpha subunit right here that's bound to GTP is going to inhibit adenylyl cyclase, is going to prevent it from making any more cyclic AMP. And then the beta and gamma subunits are going to go and they are going to activate calcium channels. So at this point, we have an inactive adenylyl cyclase, which means that PKA is not being freed to do its bullshit. And so we have functioning potassium channels, and we also have functioning, it should be inhibiting the function of the calcium channel. So yes to the potassium, no to the calcium. And so this ensures a decrease in neuronal excitability for this reason. All cell membranes have a membrane potential. And in order for electrical signals to be passed from neuron to neuron, an action potential needs to be propagated. And loosely what that is it is a increase in positive charge accumulating inside the cell. That is what propagates an action potential, and that is what facilitates neuronal communication. Now, if we open potassium channels, potassium has a huge concentration inside versus outside the cell. So, due to the rules of diffusion and osmosis, when those channels open, the molecules are going to want to move outside to where they are less concentrated. The opposite is true for calcium. Calcium is a lot more concentrated outside versus inside. So the opening of these channels would allow more calcium to go into the cell, which we don't, it's not that we don't want it, but that's what the endocannabinoids are trying to avoid right now, is they don't want positive charge into the cell. They don't want an action potential to be propagated. They don't want communication. Internalization of calcium is also how neurotransmitters get released. So by blocking the calcium channels, they're also directly preventing the release of more neurotransmitter. And this is all thanks to that G protein couple receptor decreasing cyclic AMP, and also in the case of the beta gamma subunits, directly preventing the function of ion channels. So this is take a good look, because this is just exactly what almost all endocannabinoids are doing inside our body. Now, and like I said, but both CB1 and CB2 work like this, but they do different things not just because of their location, but because of who is binding them. So that leads us into our first endocannabinoid. This is anandamide, or AEA. I forget what AEA stands for. It's really, really long. But this was the first endocannabinoid discovered. It is the functional analog to THC. So THC, of course, is exogenous. So they functionally are very similar, and they do incredibly similar things in our body. Hence, that's what an analog is. And some of the functions of anandamide are pretty synonymous with some of the basic endocannabinoid functions that I mentioned before. But I'm definitely, when it comes to anandamide, I'm going to be isolating memory, anxiety, and the runner's high. It is a partial agonist to both of the cannabinoid receptors, which basically means that it produces like a positive response, but not quite at the level that a full agonist would. A full agonist would produce like 100% efficacy and response from the receptor. So interestingly, anandamide is just it's only a partial agonist. And as you can see, it has a much higher affinity for CB1 versus CB2. These this is their Ki in nanomolar. So the lower number actually means more affinity, because that means that less of the molecule is required in order to produce a strong response. So 78 nanomolar is significantly stronger than 370 nanomolar. So because it has such a higher affinity for CB1, we can kind of extrapolate that anandamide is really, really important in our brains. So we'll go into memory first here. There are high levels of anandamide and CB1 receptors in the hippocampus. So this is where memories are encoded. They are created, and then they're stored for just a short amount of time. They memories will get sent elsewhere for storage. In the hippocampus, it's more about encoding them, and then it will also consolidate information from short-term memory for long-term storage. Again, the long-term storage is not occurring in the hippocampus, but it is more preparing the information from our short-term memory, from our working memory, procedural memory, and prepping it for long-term storage. Long-term potentiation in the hippocampus is what facilitates the creation of certain types of memory. Now, long-term potentiation is something that, of course, it involves glutamatergic signaling, it involves excitatory signaling, and it is the increased and long-term stimulation of certain circuits. It is the opposite of long-term depression. Anandamide and THC both inhibit cyclic AMP production within certain areas of the hippocampus, so they therefore decrease the calcium current in those neurons, and they prevent this sort of long-term potentiation from happening. This really acts to impair working memory, which is it impairs working memory, which is a type of short-term memory that involves like usually things that you just learned, and they're not particularly strong, and they're very short term, but they're very, very readily accessible. So anandamide and THC decrease communication in those areas and impair that type of short-term memory, that working memory, which is why if you smoke a lot of weed, you might forget that you already put your hot pocket in the microwave. That might escape your brain, but you probably won't forget, like, lose any core memories from your childhood about, like, shitting yourself in class. Like, those are gonna stay there. It's really just working memory that anandamide and, by extension, THC seem to impair. Um, I love that. I love that freaking. That's that's relatable right there. Okay, now the amygdala. This is another brain structure that is involved in. It has memory functions, but specifically, it encodes memories that are somehow attached to certain emotions. So they're associated with more emotional events. The amygdala is also involved in aversive learning and behavior. So, um, cluing you into things that you don't like and that you want to avoid. Um, and it is also involved in fear conditioning and extinction. So, fear extinction is essentially uh, if you are getting consistently stimulated by something that's fearful, ooh, and having your reaction get dampened over time, uh, and at some point, maybe forgetting the memory that had to do with a specific fear. That's fear condition. That's fear extinction specifically. So a decrease in reaction to a fearful stimulus. Um, we need the amygdala to mediate those types of behaviors, um, and anandamide um, acts through cannabinoid receptors on both GABAergic and glutamatergic neurons in the amygdala in order to kind of mediate. I know I use the term mediate a lot, but it's really just the best word. Uh, it's the best way to describe this because it's not always increasing, like net increasing or net decreasing things here. So anandamide has a pretty complicated job in the amygdala. So in order to better understand what it does, we're going to take a look at a very extreme example involving a fearless Scottish woman. Maybe. There we go. So this is real. I'm not fucking with you. This woman has a mutation in her fatty acid amide hydrolase, or FAAH gene. She has two mutations actually. And those mutations significantly decrease the functionality of this enzyme, which breaks down anandamide. So as a result, she has a lot of anandamide. She has, I think I read that she has like 2 times the amount of anandamide coursing through her body than that of a person who does not have these two deletions. So that's pretty significant. And I think this is just in the amygdala. Just in the amygdala, this is important to know. CB1 receptors are actually more expressed on glutamatergic neurons, which is not, obviously overall not what we see. It's mostly GABA, but just in this specific area. I want to say even like a specific area of the amygdala, the basolateral nucleus, is where we see way more CB1 receptors on these excitatory neurons. So if they're mostly on those excitatory neurons, then that means that the net inhibition of the amygdala will be decreased, because remember, cannabinoids are going to decrease neurotransmitter release. In this case, they are decreasing excitatory input to and within the amygdala. So there are a couple of things going on here that are preventing this woman from experiencing fear and anxiety and pain. So the amygdala have like specific nuclei acids. The basolateral amygdala is has a specialty in those fear related behaviors and fear and aversive conditioning. Um, is going to impair the creation of emotional and fearful memories and facilitate the extinction of those memories. So that acts as a sort of protector from feeling fear and by extension feeling anxiety. Um and so basically people who have like high anxiety or maybe even anxiety disorders, they have not just like different connections in these areas of the brain, but they also just have this increased excitability, which is why um for a lot of people uh, increased endocannabinoids or cannabinoids in this area can dampen all of that extra communication and maybe even make because in anxiety you also have stimuli that are not fearful that are viewed as fearful, and anandamide can help take the fear conditioning for those stimuli out of your brain by dampening these areas of communication. It's funny because what I'm about to tell you is actually some bullshit, so I apologize for the wait because what we're all waiting bated breath for me to say is that despite despite the fact that in this specific little area we have more CB expression on glutamatergic neurons, that's just the basolateral amygdala. So I think even throughout the rest of the amygdala, we still see more on the inhibitory neurons. Uh, but it's the more efficient ones are actually on the excitatory ones, which I hate even more. It doesn't make any sense. Um, but I had to bring that up because on the outset, it would look like having more anandamide would actually increase your anxiety and your fear behaviors if you have more expression on the inhibitory nerve terminals, because then you're inhibiting the inhibition. You're disinhibiting areas that um you don't want to have too much communication because it can lead to inappropriate feelings of anxiety. But the fact that they are more efficient on the excitatory ones means, hey, don't worry, that won't happen because they're just so much more efficient on the excitatory ones that increase anandamide probably won't make you fearful or anxious because they're just so much more efficient at dampening the excitatory activity. I mean, Jesus Christ, could our body be any more of an asshole? Could it be any more confusing? Um, so I I guess that's good. Thanks, anandamide. Anyway, this woman is literally also immune to stress. Um, so we're still with this woman. She was very intensely studied, but she's immune to stress. So I'm gonna get a little crazy right now. I'm gonna get a little stressy right now because uh, I'm gonna get a little stressy, which is something that this fucking woman has never experienced, and I hate her. I'm just kidding. But endocannabinoids basically make sure that there's a normal amount of stress hormone present in our body. So they make sure that circulating like cortisol and ACTH, they make sure that those things don't get out of hand. Because chronic high amounts of stress hormone is obviously kind of bad for you. Um, so uh, the HPA axis, which mediates stress, the hypothalamic pituitary adrenal axis, uh, is kept at bay by endocannabinoids. Um, in response to stress, the HPA axis will get activated, And then the circulating stress hormones will increase the expression of FAAH, of fatty acid amyloid hydrolase, which remember breaks down anandamide. And less anandamide will mean that there will be less inhibition on glutamate release to the basolateral amygdala, and therefore anxiety and stress behaviors will occur. We call that anxiogenesis, which is the formation of anxiety. So that's interesting, right? We have endocannabinoids to keep the amount of stress hormones in check, but during stress, our body releases a lot of those stress hormones. And they actually end up decreasing the amount of anandamide and endocannabinoids. So a really nice negative feedback mechanism that we have. But this freaking lady, her FAAH doesn't work in the first place. So it doesn't the if she even has a stress response, it doesn't it's not going to decrease AEA signaling because it doesn't have anything to act on. Specifically what the stress hormones act on is the creation or is this enzyme. So if hers is already faulty, then the increased stress hormones aren't going to do anything. And because anandamide keeps the stress hormones at bay during physiologic conditions, she might not even get to a point where she has high circulating stress hormones ever. So I think that's kind of rude, to be honest. I would love to have a dysfunctional fatty acid amide hydrolase. I'm just kidding, but it is very cool. And I just thought of this as a side note. People who have like damage to certain areas of the amygdala also just do not have fear. They will indiscriminately, like, they're very sociable. They indiscriminately just like approach people and talk to them. They have no sense of caution. I don't know, just popped into my head, made me think of it. Okay, runner's high. This is everyone is familiar with the runner's high, and anyone who's been high on any drugs before knows that the runner's high it's not comparable, but it's like, it is the next best thing, unless you hate running like everyone does. Just kidding. But there is a reason why it's called a runner's high. There is an amount of euphoria that is associated with moderate exercise, and endorphins are commonly cited as the reason behind the runner's high, and that is because they on the right here we have that's actually a couple of beta endorphins kind of smushed together. And then we have the mu opioid receptor, which of course is that's where opioids like fentanyl and morphine bind, and that's how they produce their analgesia or pain relief, and also their euphoric effects. So because beta endorphins are produced during exercise and they bind this receptor, it was commonly accepted that, well, beta endorphins cause the runner's high. But plot twist, that's actually not all true. They it's actually endocannabinoids that have a much larger role in this exercise induced euphoria. And this chart actually shows how blocking either opioid receptors or cannabinoid receptors affects anxiety and pain. So if we look at the top bar, these are mice. This is the model of anxiety that they have for mice. I think they put them in the dark. I think that's what made them anxious. But in any case, the vehicle is the control basically, so they were injected with nothing. Naloxone is the blocker for the opioid receptors. This is a CB2 reverse agonist, so it doesn't block, but it literally reverses the effects of a ligand that would normally bind to CB2. We have a CB1 antagonist, so it completely blocks CB1. And then again, right here, we have a reverse agonist at CB1. So in the anxiety model, blocking the mu opioid receptor did not really change the anxiety model here. In a sense that the mice experience the same amount of anxiety, and this is still an exercise induced thing, pretty similar to what they did in the controls. Whereas blocking the cannabinoid receptors did still, even with the blocked receptors, wait, I have that backwards. Oh my god. I'm gonna have a sip of water. And the water will give me your brain. Okay, here we go. Opioid blocker did not change anxiety, but blocking and reversing the cannabinoid receptors caused more anxiety in the groups that were made to be anxious after exercising. So they're basically testing if the mice still got a runner's high, if they still experienced anxiolysis even with the these receptor blockers, and they determined that they still didn't experience it with the opioid blocker, but they did with the cannabinoid blockers. So now we'll go to the pain model, because that is another aspect of of the runner's high and of beta endorphins, is you feel less pain. I mean, it's not extreme, but like you just, it kind of, if you're like a runner, especially like if you're a marathon runner, it can kind of keep you going because your um legs aren't getting as crampy and tired, what have you. So when they tested this model, this aspect of the runner's high, it was kind of the same thing. When they blocked the mu opioid receptors, it didn't change, whereas blocking the endocannabinoid receptors um did not uh result in any sort of pain relief. So what this shows is that literally the runner's high will get blocked if we block our cannabinoid receptors. It will not get blocked if we block our opioid receptors, which outlines the importance of endocannabinoid in the runner's high, and also kicks beta endorphin out of the frickin' club here. Fuck beta endorphin. I'm just kidding. I'm sorry. There are obviously a lot of other processes related to anandamide in concert with other molecules, but we're gonna take a break from all that. We're gonna take a break from frickin' anandamide. We're gonna go to 2-arachidonyl glycerol, or 2-AG. All right. 2-AG is our body's analog to CBD, the other hot topic phytocannabinoid in the cannabis plant. And its affinity for CB1 is not as high as anandamide, but it is more efficacious, making it a full agonist at both of the cannabinoid receptors. So I think I slightly mentioned this earlier, but just to reiterate, if something is a full versus a partial agonist, that means it has a higher efficacy. So it produces more of a response even at lower concentrations than the partial agonists. So it's kind of interesting here, especially since it definitely has a freaking higher affinity for CB2 versus anandamide, that's for sure. So I'm going to be discussing 2-AG's roles in the body as a function of CB2 receptor expression, basically. So that's going to be, we see its expression in the GI tract and in B cells, as well as microglia. Um, so let's take a test. I keep hitting this. Freaking THCD's nuts. Got him. THCD slides are so wonderful. Oh, yes. Okay. Uh, so plot twist. All of these are technically the immune system. I freaking kind of lied to you. Even the GI tract. Because within the GI tract, 2AG will interact with CB2 as a way to kind of protect us from our gut flora, and also protect the gut flora from being attacked by our own immune system. Um, sort of a twofer. Um, and CB2 is also and this is kind of a theme that we're going to see with 2AG and with CB2. But CB2 is gets upregulated during pathological states. So people with inflammatory bowel disease or IBS also will have an increase in the expression of CB2 receptors in the GI tract. Um, and it's kind of a compensatory mechanism, because there's all this inflammation happening in the GI tract in these sort of disease states, and so our body's way of compensating is like, well, let me upregulate the expression of a receptor that's going to mediate anti-inflammation. Um, so and this is in their epithelial cells mostly, not really in the nerve terminals like in the enteric nervous system, but actually in the epithelial cells of the GI tract. It's new water. New water, new water, brand new water won't poison me. New water and new mic, which I'm very excited about. It's so new that I keep forgetting it's there though. Whew, okay. Um So yeah, remember that. CB2 gets upregulated in pathological states. Um so uh oh yeah, compensatory mechanism. God, my handwriting. There we go. Okay. So the big takeaway already is that CB2 and 2AG have a pretty significant anti-inflammatory role in the body. If we move on over to B cells, um, they're part of the adaptive immune system. They make antibodies and they present antigens, meaning that they um, sample our lymph and our blood, and if there's something fishy, they process it and then they show it to another immune cell that will then sort of mount a response against the antigen if it is indeed something that the body deems foreign and dangerous. Um, in the spleen, there is an area of there's a population of B cells in an area called the marginal zone, um, and they sort of aid in the defense of certain defense from certain bloodborne pathogens. So in this area of the spleen, again, there's kind of sampling the blood, looking for antigen, making sure that there's no gross things there. Um, and it's 2AG that is responsible for uh, attracting those cells to the marginal zone. So this is interesting. In this case, it's not really that 2AG is acting as a neurotransmitter, but as a what we call a chemoattractant, a chemical that um, attracts certain maybe other molecules, but in this case is attracting cells to a specific area in order for them to carry out their function as defenders against these bloodborne pathogens. It's very, very crazy. No problem. I will take any question. Even if I said it 1,000 times, I'll freaking take it. but I did not say this 1,000 times. It is naturally found in the body. It is one of the endogenous cannabinoids that our body just naturally makes. And it is an analog of CBD, so it's comparable to CBD. The other one that I was talking about earlier is that's comparable to THC is anandamide. So 2AG more or less is the CBD inside of us that our body makes. I love it. Um, and unfortunately, I talked a lot about anandamide, but 2AG, I wish there was more. There's just not as many like studies out there about like its actions other than the immune stuff. But I know it's still there's still a lot of 2AG in our brains. Maybe the real high was the 2AG inside us all along. Hell yeah. Oh my god. Ah, so useful. Makes us like high, but also prevents immune overreaction. It's just a jack of all trades. Um, so yeah, very interesting. It attracts B cells to where they need to be doing their job. Um, but also there's this kind of interesting it's a little bit of a contradiction here because in this case, I'm talking about how 2AG helps mount an immune response by bringing the B cells to where they're supposed to be. However, um, in certain types of B cells, um, well, this basically happens when like we're sick, and immune response is happening. CB2 goes up as it does during times of during our pathological times. Um, but when the B cells get activated during those times, they produce a lot of 2AG. And the 2AG actually inhibits the B cells that are producing it in a negative feedback kind of way. Um so this again demonstrates this very like regulatory role that 2AG and endocannabinoids have in our body, where um it's helping us kind of mount an immune response, but at the same time, it kind of checks our immune system and makes sure that it doesn't, that it plays nice, that it doesn't, get too crazy and actually end up hurting us. It's crazy shit. And we see honestly, the biggest example of this in microglia. Microglia are so glial cells in general are just basically support neurons. They're support for the CNS. You may have heard of like oligodendrocytes or astrocytes and stuff like that, but microglia are they provide immune defense for the CNS, like a little personal immune defense for the CNS. And they're not they don't have a lot of CB2 expression, again under physiological conditions, but once we get pathological, like if there's a spike in inflammation, then we also see a spike in that CB2 expression. And again, 2AG will act through CB2 to prevent microglia from going fucking sicko mode on our CNS. Because microglia can become very cytotoxic. They can kill cells like super easy, and they can release inflammatory mediators that can end up like significantly hurting us. So 2AG steps in to be like, hey, maybe don't do that. It prevents the release of those inflammatory mediators and the release of what's it called? Free radicals. Free radicals are also used in immune signaling, so 2AG will prevent glial cells from overreacting to invaders. So very cool stuff. Very cool endocannabinoid effects on the immune system. we're gonna backtrack a little bit because what have we not talked about yet? It's time for the phytocannabinoids. Um, we all know and love them. They come from the cannabis plant, and unlike our endocannabinoids, the plant makes these molecules as a way to deter predators from consuming them. That's why edibles can taste like dog ass. Unfortunately for the plant, it didn't consider that we might set it on fire and inhale it to get high. Um, the bulk of the endocannabinoids are I think you can kind of see on this nug, hopefully. There's a lot of those little crystal things. Those are liddy trichomes. They produce the bulk of the cannabinoids and the terpenes. And in order to actually experience the psychoactive effects of THC, you have to heat it up to decarboxylate it to remove that carboxylic acid group. Is it gonna let me this right here? If this is attached to it, it is not psychoactive. And same with CBD, even though CBD is not psychoactive, it still undergoes decarboxylation. And I believe that it does need to be decarboxylated in order for it to have any of its like therapeutic effects. So it also needs to be decarboxylated in order for you to like be able to eat it and get high too. That's why people will actually bake their plant material before cooking with it. And then because cannabinoids are lipophilic, they like fat. You actually it actually needs to be in a fat like oil or butter in order to be bioavailable to you. Since CBD is lamer, let's talk about it first. I'm just kidding. I love CBD. But its action in the body for whatever reason is like it's just not as well understood as some other cannabinoids out there. In fact, there's literally I couldn't even find anything on its cellular mechanism, which made me cry on the inside. But I can still sum some of it some of the functions up. So since CBD and 2AG are analogs, we know that CBD is mainly going to act through CB2. And we just learned all about what CB2 does in our body. So that's how we know that CBD is just a nice, decent, non-psychoactive treatment for certain types of inflammatory pain. A lot of CBD's therapeutic potential comes from its interactions with other receptors that have nothing to do with cannabinoid receptors. So epilepsy is very commonly treated with CBD. And remember that GABA produces inhibitory effects. So when CBD agonizes one of the GABA receptors, the GABA A receptor, this is going to dampen neuronal excitability, which is what we want in epilepsy, because epilepsy has to do with really discordant and too frequent electrical signaling and communication within the brain. So this is a way to kind of put a stop to all that. There are certain psychiatric illnesses that can need to move. Okay. Certain psychiatric illnesses that can be treated with CBD potentially, like anxiety, mood disturbances, depression, and CBD actually binds one of the serotonin receptors. It binds the 5-HT1A receptor, which leads to a relief of anxiety as well as depressive symptoms. Um, and this is, of course, because, it's not just one neurotransmitter binds a receptor and then boom, we're done. The 5-HT1A receptor actually modulates other neurotransmitters like dopamine and norepinephrine. Um, Dopamine and norepinephrine, as a result, they actually get released in certain areas of the brain that can aid in antipsychosis as well as possibly schizophrenia. Um, it increases dopamine and norepinephrine in the medial prefrontal cortex, the hippocampus, and I want to say the striatum. Now, in a lot of mood disorders and schizophrenia, um, with dopamine interaction, you have areas of the brain where you have too much and areas of the brain where you have too little. So clearly, CBD's ability to increase dopamine in these areas and having it have a positive effect means that, for example, for schizophrenia, it might actually decrease hallucination symptoms, or it may, because especially with the prefrontal cortex, I'm sure it improves like affect. Um, Certain mood disorders and schizophrenia have blunted affect, so there's not any, what's it called, expression of emotion, but there's also like a lot more to it. But these are like very low dopamine things. The blunted affect, so increasing dopamine in the MPFC can help with that specific symptom of schizophrenia. Um, because if we're increasing I'm just trying to say, if we're increasing dopamine in other areas of the brain, it might not be helpful for schizophrenia. Like if there were areas of the brain where it increased dopamine, people be tripping because of serotonin and dopamine. So if you're increasing it in a specific area, you might be worsening hallucinogen symptoms. So I just personally think it's interesting that, like, CBD increases dopamine in apparently the exact right areas. Um okay, well, chronic pain. How could I forget? So, the CBD, of course, also mediates anti-inflammation. That was one of the common things that people would come into my dispensary for, was it was sleep, but also pain. And there was always a question of whether it was chronic and whether it was like, it seemed to be inflammation, because if it was, then CBD was definitely the better choice to sort of treat those things. Um, and one of the ways that CBD can help relieve pain is by blocking these specific receptors that are actually cation channels called TRPV1. Um, and they are they're in a lot of different places, but there's ones in the spinal cord um, that mediate pain signaling, specifically like heat pain, like pain in response to like burning yourself. Um, but sometimes pain, there are people who like, I think fibromyalgia, where it's like you're in pain but there's nothing stimulating it. So I think the idea is that CBD, whether or not there's real pain signals that are getting to you, CBD can help dampen those, specifically through this nice little cation channel. We love you. Okay, main event. Well, I don't know. I like anandamide, but let's talk about THC. Um, the chemical that people tattoo to themselves, is the main psychoactive component of the cannabis plant, and tolerance is, I think, a lot of the biggest like complaint about THC is the development of tolerance. And I wanted to bring that up right off the bat because of we've been talking about receptor interactions. THC is less efficacious than the endocannabinoids. So it's even more partialer of an agonist than anandamide even. And for that reason, a very normal thing to happen when you have receptor binding is that it internalizes so that you don't this is to kind of prevent overactivation of certain receptors. So since THC has I believe a higher affinity for these receptors but less efficacy, you get more internalization, more downregulation of the receptor. So pretty soon the amount that you were ingesting before does absolutely nothing. Thanks, efficacy. Okay. So the high. Like almost all drugs, all psychoactive drugs, uh, THC increases dopamine within the reward system. And that is what creates the feeling of being high. That is what creates the euphoria. I have just this is just a general kind of picture here, but the VTA is where dopamine is made. So I'm not really talking about that area, but certainly the nucleus accumbens and the well, let me draw the striatum. So it's like this long. It's not the corpus callosum, but the striatum is kind of this long thing, and the NACC is a part of it. That area in particular has to do with self-administration of drugs, addiction potential, motivating behavior as it pertains to getting drugs and stuff like that. So, like any other psychoactive drug, THC does release a good amount of dopamine into this area. And so cannabis is addictive, just like literally anything else. But the reason why people talk about how it's not as addictive is because it doesn't seem to release as much dopamine into these areas as other drugs. Like cocaine and heroin are like they release they turn your whole brain into dopamine basically. So it is true that cannabis doesn't have that significant of an impact on dopamine production in the reward system, but it's still there, and so it still has an addiction potential. Speaking of pathways, I love that. Oh my god. The best is when they turn to the camera and say it. It's like oh my god. They're like Talladega Nights. I don't know. That was the first movie I could think of. Oh my god. Uh I'll do it. Okay. Despite its low addiction potential, THC still shows increases in dopamine. It's so good. At this point, it does feel. It's like when you say your own name, and it just feels weird. It is like that now. I love it. Oh my god. But speaking of pathways, because reward pathway, uh, there, oop, whoa. God, my handwriting. Oh, here we go. Pathways. The pathway of THC is very different when you eat it versus smoke it. When you smoke it, basically, a lot of it doesn't necessarily get broken down like metabolically. It gets stored in your fat cells, which is why THC can stay in your system for a very long time because there's fat everywhere on your body, and it just kind of hangs out there. But when you consume edibles that have THC, um, your liver does this really really goaded thing where it adds a hydroxyl group to the THC, turning it into 11 hydroxy THC. It has a much higher affinity for the cannabinoid receptor, CB1. It's probably the same amount of efficacy, but because it is so much more, it has such a higher uh, affinity for the receptor. We think that this is why highs from edibles tend to feel more more intense. And this is an oh my god, it's loading. This is an educated guess right here, but I would say that 11 hydroxy THC having a higher affinity means that it kind of opens it up to be able to bind to more areas um of the brain, basically, because if it's the same amount of efficacy, then it's not like it's binding and then oh my god it's so much stronger. It's just it's able to displace anything else that would bind the receptors, any of your endocannabinoids, any other molecule, because other molecules can't in your body can bind cannabinoid receptors, but because of the ridiculously high affinity of the 11 hydroxy THC, those things just don't stand a chance, and that's why your high is longer and more intense when you eat edibles. Um, and also why edible tolerance is separate. People have found than smoking tolerance. Like, I feel like I found that. I used to just smoke all day, every day, forever and ever and ever. But always my edible tolerance, I'd have like 2 to 5 milligrams, and I'd be like, okay, we're gonna stay here. Like we don't need to go any more than that. um, while we're on the subject of eating, let's look at the most famous side effect of THC: eating. Um, this was really cool. I liked learning about this. um, this info is brought to you in full by people who got rats high for science. Oh my god. So we have this thing called the hypothalamus. And inside the hypothalamus, we have this thing called the ARC, or the arcuate nucleus. Gotta get that rat high. I mean, it's for science, you know? We gotta get them litty to get that info. The the arcuate nucleus regulates our hunger and satiety, mainly through these two populations of neurons. We have a agouti-related peptide, which increases appetite and decreases energy expenditure, and pro-opiomelanocortin, or POMC, which promotes satiety and decreases appetite. I don't know why it's called a agouti-related peptide. I don't know. I don't understand. I don't know why they did that. That's an agouti. That's a agouti-related peptide. I think they look the exact same. So maybe that's why. In any case, these two populations of neurons work against each other. They are opposites, and they are also affected by circulating hormones. So, for example, ghrelin, that is actually the hunger hormone, is produced by your stomach, and in response to increased ghrelin, or increases of that hunger hormone, it will activate the AGRP neurons and mediate feeding behavior, food seeking behavior. Leptin is the complete opposite. Leptin is anorectic. Anorectic? One of those. It decreases appetite, so it inhibits those AGRP neurons, and it stimulates POMC neurons, which increases the feeling of satiety. There's also no one would tell me where it was, but it's like the most important part here, but there's just a random fucking who knows GABAergic input to the AGRP neuron. Oh, it's from the dorsomedial hypothalamus. Dorsomedial hypothalamus. DMH. That's what has the inhibitory input to the ARC. Okay. That was mostly for me. I was sad. So this GABAergic input comes from another part of the hypothalamus. and it synapses onto just I think just the AGRP neurons. Uh, and when you smoke weed, the THC is gonna bind what the heck? Okay. When you smoke weed, the THC that you inhale is gonna bind on the nerve terminals of that inhibitory neuron. There are CB1 receptors present on that neuron. So the THC will bind them. And what happens? Retrograde signaling, endocannabinoid signaling. What happens when we bind on nerve terminals? We lower the amount of neurotransmitter being released. In this case, because the neurotransmitter is GABA and it's inhibitory, we are disinhibiting the AGRP neurons. We are telling them, eat, we are not satiated. There's significantly a significant decrease in the inhibition of these neurons, thanks to THC. And it will actively prevent the feelings of satiation, because up here, like, we could see right here that the AGRP neuron is directly inhibiting the POMC neurons right here. So they're releasing less of their own satiety in promoting neurotransmitter. But also up here, AGRP is inhibiting, with honestly not even just AGRP. These neurons are also releasing their own GABA transmitters and neuropeptide Y, which is another hunger promoting neuron. So you just get this freaking wave of all of these hunger promoting, satiation preventing signals in your brain face. And that is why you eat so much. That's how we know, the munchies are in your head. But yeah, this is my favorite. This is my favorite thing to study, my favorite effect. Now, okay. I said earlier that I super forgot to talk about embryo implantation. I forgot to put that in the presentation. So I want to, oh my god. I think I watched one episode of their show. It's pretty funny, but still, iconic duo. I want to really quickly touch on embryo implantation because the placenta releases anandamide, of course, which is the THC analog that we produce. And it looks like increased amounts of anandamide are associated with spontaneous abortion and poor, like, kind of like, not like poor I guess poor implantation, yeah. But on the flip side, lower amounts of anandamide are also associated with poor implantation even before. So like high amounts of anandamide, it seems like there will be a formation of the placenta, but it just ends up not being all that viable. Whereas with anandamide that is too low, that's more of like a miscarriage thing. Like there's no like latching of the embryo in the first place and no viable creation of the placenta. So, there's been scientific talk about women or anyone who experiences spontaneous abortion. Maybe they require more anandamide in their myometrium, which is the muscular part of the uterus, and then those who like have more later term problems with implantation or with the placenta might benefit from inhibitors of some sort. So, and it's always an interesting kind of pregnancy with anything is always an interesting conversation, because I would certainly have pregnant women come into the dispensary buying things for themselves. And I never really thought to ask about, like, because I'm this is like a fucked up thing, but people were really like, they'd share pretty much everything with us, so it wouldn't have been inappropriate for me to ask, "Hey, did you hear about the implantation stuff?" Because maybe they were there specifically for that. Um, but yeah, that would have been interesting. My I wish I didn't forget to include that in my presentation, so I could have included some more numbers on the subject, but yeah, this has been so very great. I did not have my co-host, my producer here with me today. Poor Amy. She had to work late, very late. So, hearts out to her. She will be so proud that I set up this microphone all by myself. Ha 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 ha. And yeah, I think the only thing that I didn't get a chance to touch on was sleep. Um that is a very, like, sleep is pretty complicated. Especially since, like, on the outside, I would generally only recommend that people like smoke and get really high for help with sleep. I'd be like, don't do that every night, one, and two, if you are super anxious, I would not, because like I would have to ask people, like, what's keeping you up? Are you anxious about something? Because if you are, you should probably try CBD instead, because that is going to be better at, like, dampening the weird communications that are probably going on in your amygdala and your hippocampus. Whereas THC, because it's psychoactive, might just keep you up, and you like, if you're the kind of person who stays awake because you're like, then you know, THC might not be the thing for sleep. But that's the thing. I didn't get a chance to uh study it in depth. I would have loved to see that. I would have loved to have been proven wrong, honestly, cuz that's just always been my basis when it comes to when I was a professional weed smoker and had to recommend things to people. Stoner science. I am happy that the majority of this was science, Like, it's oh, that was another thing that I wanted to talk about with the eating thing. This will be the last thing. People, a lot of times, if they are chronic weed smokers, they will get into this thing where they literally can't eat unless they smoke weed. They literally cannot eat things unless they smoke weed. That's probably, like, the biggest side effect, like, the biggest unwanted side effect of, like, weed withdrawal is not being able to eat. Um, and I was reading about how, like, if you have chronic inhibition of, these GABAergic neurons to the agouti-related peptide neurons, um, then you're essentially, I mean, you're changing the landscape of all areas of your brain, but specifically in this area, um, your body has become more dependent on a pathway wherein it relies on neuronal input rather than hormonal input, is, like, kind of what I was seeing. So, like, normally you have these hormones to kind of increase or decrease your satiety, um, but you're kind of you're putting more weight into a completely different pathway. And so even if you have maybe normal circulating amounts of these hormones, they're not going to make you hungry because your brain is relying on disinhibition of this freaking GABAergic neuron from the DMH that is not getting if you are not smoking weed. So, fun stuff. It is possible to get out of that, but I think it's that and nightmares are the biggest issues with cannabis withdrawal. Well, I hope everyone enjoyed. I hope everyone learned one thing or another. I hope everyone likes my picture of the agouti, a animal that I did not know existed. And yeah. Whoop, whoop. Thank you, my friends. I will have the sources and a vote for the next subject out later this week, probably.